Learning about the electricity of a cell uses lots of very specific terminology that's used in a physics class. Whether the electricity is in the human body or whether it's in a car battery, it's all about the movement of ions. Throughout this course, we have been and will continue to be constantly talking about sodium ions, which are really common outside a cell. And we will also be usually talking about potassium ions, which are very common inside the cell. This distribution, though, it's not balanced in the way that I drew it. It should be like that. It's not balanced because inside of the cell there are tons of proteins that carry mostly a negative charge, making the inside of the cell negatively charged. The difference in charges is called the voltage. And when I say charge, I mean, yes, I do mean like literally, if you were to add up all of the positives and negatives inside versus outside, that would be the measurement or the numerical value of voltage. So right here is why I don't like talking about voltage, because you have to go to two places to get a reading. I don't like that. If we had an electrode inside a cell, here, let me draw you an electrode. If we had an electrode inside the cell and then another electrode outside the cell, it would be the difference of these two things that would give us a reading of about negative 70 millivolts. And we're going to use negative 70 millivolts as an example many times in this class. This means that when everything was added up, we had 70 more negative charges than positive charges. Actually, that's not true. It's a log scale, but it's perfectly fine for our uses here. Here in human A and P, we don't call this the voltage. We call it the potential, and that the potential is measured in millivolts. So it's like us saying this is length, and we measure length in meters. So potential is measured in millivolts in cells. And we're going to learn about a few different types of potentials. So, I mean, why do we use this word potential? Why don't we just call it voltage and electricity? Because the difference in charge, the numerical voltage, is an indicator of the ability to move ions. A cell membrane is separating these charges from each other. But if some channel in the cell membrane would open, then we're going to see movement. And that's why we call it potential, because there is a potential to be moving ions. The resting potential is the numerical voltage at which a cell sits when they're in homeostasis with their environment around them. Actually, it's probably more accurate to say when they're maintaining their resting potential, they're doing that in order to maintain homeostasis with the environment around them. As we've just learned, voltage is a separation of charges. So when a cell is at rest, the cation-anion distribution exists as you see it here. There are tons of sodium cations in the interstitial fluid, and there are a lot of potassium cations in the intracellular fluid, but they're not as densely packed as sodium. There's way more sodium than potassium, always. However, the inside of this cell is more negatively charged than the outside, not just because of the proportion of sodium and potassium, but because, remember, a cell is constantly making proteins, and those proteins are usually negatively charged. Further complicating this situation is that there are leaky channels through which the sodium is escaping into the cell and the potassium is escaping out of its cell. The sodium channels are open to sodium, and it allows sodium to flow along its concentration gradient using facilitated diffusion. And this makes sodium diffuse into the cell. But there are also potassium channels that allow potassium to escape down its concentration gradient. The movement of these two ions constantly disrupts the resting potential, right? As sodium moves out, sodium moves in, we gain positives. As potassium moves out, we lose positives. And so this leakage of these ions disrupts the resting potential and moves it away from the value of -70, which we're going to use as optimal. Luckily, there is this protein, and I have pictures of two of them here. There's a protein that is called the sodium-potassium pump. We can abbreviate it in our notes as the NAK pump. This pump uses active transport using ATP to transport sodium and potassium back to their original position. You need ATP to do this. You can only do this if you have energy. Also, the detail of this diagram, check this out. It shows you one of these sodium-potassium pumps moving sodium, and it shows you moving 3 sodium cations, right? The other sodium-potassium pump here, it shows you moving 2 potassium cations. This is because sodium leaks into the cell at a much faster rate than potassium leaks out. So for every 3 sodium cations that invade the cell, there are only 2 potassium cations that are escaping. And so this is why the sodium-potassium pump, it doesn't make an even exchange. Every time it pumps 3 sodium cations, it only pumps 2 potassium cations. There are many types of potentials in the body. The word potential just means electricity. Probably more accurately means the movement of electricity. But no matter what you want the definition of potential to mean, it's a value of voltage. And we will experience and come to know 3 different types of potentials. A resting potential, which we've already discussed, is going to be for us -70 millivolts. And it's the potential or voltage at which the cell is when they're performing homeostatic processes. Just as we saw with the osmosis lab, a cell that's isotonic to its surroundings, it doesn't shut down exchange with its surroundings. It continues to exchange with its surroundings as needed. So a cell sitting at its resting potential, it's not shut down from voltage. What it's doing is it's continuing to exchange with its surroundings. A local potential, which can also be called a graded potential. It's a stimulus that it doesn't go anywhere. The best analogy I have for this, it's like trying to light a lighter that doesn't light. You get it to get a spark, but you don't really get a consistent flame. Right now, there are many neurons in your brain that are having local potentials that produce no conscious thought, no involuntary output, nothing. Nothing. It's a lighter that's not lighting. These potentials that die out are called local potentials. So you've excited a neuron in some way, but you've not created enough of a change in voltage to move electricity from point A to point B. An action potential is something that we've mentioned at various times in this course. An action potential is the movement of electricity from the cell body of a neuron over the axon hillock, down the axon, and all the way to the tip of one of those axon terminals. So as an action potential moves down the axon of either of these neurons, really, it is allowing sodium-potassium ions to be exchanged in an incredible magnitude at every single one of these locations. Right now, there are local potentials happening in your cell bodies of your neurons in your brain. And if those local potentials, let's put local potential right here, if it creates enough voltage to actually go over the axon hillock, then it will be able to propagate that voltage down the axon. And I just want to label this area here as the axon hillock. It is an area of resistance, right? So a full thought goes over the axon hillock. Another thought, it does not. If the local potential, if it doesn't get enough voltage, then it won't go down the axon and the thought will never be completed. In an action potential, we use this term propagation, and it's a term that indicates conduction of electricity, but conduction within a human tissue. If we were in a physics class, we might just call this conduction to indicate the movement of electricity from point A to point B. Think of this like when you plug in a lamp. There's conduction from the outlet to the light. Electricity flows from your wall socket to the light bulb. That's the same thing as an action potential. Carrying electricity from your brain to the muscle in your big toe. And instead of like waking up your big toe, what it really does cause is a contraction. These types of channels were discussed in a previous chapter, and now they come back at us with a vengeance. The channel in the middle of this picture, the purple channel, is a leak channel. It is always open. It is incapable of closing. If a substance fits into it, that substance can move through it, and substances will only move through it according to their concentration gradient. In this picture, you can see all of the little purple dots on the top of that cell membrane there, and then not so many of them on the bottom. So these little cations or ions are moving according to their concentration gradient from high to low, but they are using that leak channel to move across the cell membrane. The blue channels that are on the left are what are called ligand-gated channels. There is one with the channel open, and then there's one with the channel closed. These ligand-gated channels, they only open when a neurotransmitter connects with them. In this picture, the neurotransmitter is that orange sphere, both of them, that are connected to the ligand-gated channel. A ligand-gated channel, a ligand is like a key that opens up the channel. But the ligand itself, that yellow orangey sphere, it never moves through the channel. What moves through the channel for us in this class is usually sodium and or potassium. But note, no matter what, the blue ions are moving through the blue channel according to their concentration gradient. So again, the channel is not determining this movement. The green protein on the right, or the teal colored one, it's a little bit more complex than what we have previously seen before. And this is a voltage-gated channel, which we encountered briefly on the axon terminal of a motor neuron at the neuromuscular junction. A voltage-gated channel opens when there is a change in the resting potential. So a ligand-gated channel opens when there's like a physical key that gets put into the channel to open it up. But over here, with a voltage-gated channel, we have to generate some electricity to open that up. I don't know if you know this, but many hotels that use key cards, what they do is they have a magnetic strip that actually runs through the door, and I'm not explaining this really correctly, but to the best of my understanding, you insert the key card and it breaks that magnetic strip, opening up the door. So it makes a change in voltage, which then allows the door to open. And this is why the door handles kind of feel warm, because there is electricity running through them.